All right, so let's start with the module 7, cellular interactions, and we'll see that very straightforward chapters. For the other chapters, we essentially talked about the cell in isolation, what they're doing, and now we are talking about how the cells are interacting with each other. We talked about how the cells actually receive signals, and now we are talking about that when they actually act together in a tissue, and how these cellular interactions actually takes place. So these are all straightforward, and this will be the first very important slide that know them all. So just keep that slide handy. So we are going to talk about the occluding junctions. Means that, again, as the name suggests, it occludes. It just doesn't allow things to pass through them. And in addition to that, it acts also to keep the cells together, the epithelial layer of cells, because sometimes if you don't, then the top layer of the epithelial cell layer, although they slough off every day from different areas, but it will be again coming off the basement membrane much more easily. So we're going to talk about that. Then we are going to talk about the anchoring junctions. And the anchoring junctions are just below the occluding junctions usually. And so this is very important. This is in between lines, and know this very well, difference in between that. All anchoring junctions, which are actin based, they are called adherence junction. So your cell to cell, as well as cell to matrix, if actin is involved as the microfilament instead of intermediate filament, we will always call them adherence junction. Now, the adherence junction is two types. If it is cell to cell, we call this as the adhesion belt. If it is a cell to matrix, which is usually most of the times the basal layer on which the cell is resting, we call this as a focal contacts. So these are the differences, but both of them act is involved. On the other hand, if the anchoring junctions are made of intermediate filament, we actually do not have a common name. Maybe desmosomes for each one of them, but the cell to cell is specifically just desmosomes. So instead of adhesion belt, here it is cell to cell is desmosome, and the cell to matrix, instead of focal contact with the actin, you have hemidesmosomes or hemidesmosomes. And the difference between that, you just can think about that intermediate filaments are stronger, and usually there are much more larger protein plate-like proteins are involved in the intermediate filament junctions, and these are actually much more stronger as such compared to the adhesion belts and the focal contacts. So it is much more difficult to remove or take the cell out with the desmosome or hemidesmosomes. So just you can think about that. These are just simple screws or nails, and these are just like really plated bolts and nuts and bolts. You can just think about in a very crude way like that. These are much stronger. And then we are going to talk about the communicating junctions, which are the gap junctions. And this will be a very small part. And mainly this doesn't have any structural role as such. This is just only for communication, mainly for electrical communication. We're going to talk about that. All right, so this one is important. So let's take a look at each of the junctions, and we'll just go over each one of them, and particularly not in this order. So we will go just like the order from the previous slide, this one. So we will start from the occluding junction. So if we just look at the occluding junction right here. So before we go in there, let's take a look at what we are essentially seeing. So what we are seeing right here is a basal lamina right here. This is a laminar layer, and sometimes you have heard the word, and you may be curious about that. We say there's a basal membrane, and the basal membrane is actually made up of two layers. Do not have to go into the detail of that. And this is a laminar layer right here, which is the basal lamina. This is the top layer. And this is, you can see that there are a lot of fiber in here, and this is a reticular layer. So this reticular layer and the basal lamina actually together actually make it. Let me just share a Google. So here you can see that we are just, you have these epithelial layer, and see right here, this is a lamina propria, or the reticular layer that you are seeing. And right here, we are calling this as a basement membrane. And and the top right here, and you have the blood vessels and all those kind of things. And right here, this top thin line that you have, this is essentially the your basal lamina or the laminar layer. So you can see that right here, this is essentially the basement membrane, do contain this reticular layer as well as the basal lamina. So this basal lamina, this pink line, we are only considering this because this is the layer, this pink layer is on which the epithelial cells are essentially lodging onto. I'm just trying to see if there is this is probably the most complete picture. So if you just look at the entire layer, you see that this is a reticular lamina right here. You have a reticular layer. Then you have a lamina densa right here. Lamina lucida and densa, these are the top two layers which actually make the basal lamina. And you see that hemidesmosomes, and we are going to see the focal contact, they only interact with this lamina lucida layer. They don't even go onto that. So just the top layer of the basal lamina is involved. But this detail is absolutely not necessary. Just in case that if you just curious, what is the difference between a basement membrane? Because you see that word all the time in biology, difference between the basal lamina and the basement membrane. All right, so now let's take a look at this, that you have an epithelial cell and they are polar. That means that they have an apical surface right here. And if this is intestinal lining, cell of intestinal lining, they have all these villi, microvilli, and this is the apical surface. Then you have the lateral surface, and then you have the basal surface on the basement membrane. So first, we are looking at the tight junctions. So the tight junctions are keeping these cells together, and also the main function, of course, they bind the cells together. That is true, but it is essentially occluding the space on this side as well as on this side. So here you can see the cell number. This is the central cell. This is the left side. This is the right side cell. And right here, left and right. And of course, remember that this cell is three-dimensional. So it is all side of the cell. It is just it's not a 2D picture. It should be all these junctions are essentially 3D pictures. So we will see some pictures with that. So it's all around. And what it is occluding, this basal and the lateral area from the apical area. And we know from our previous chapters that there are two reasons for that. The first is that the environment in the apical surface and the basal surface are different, and they do not allow free passage of ions and material in between the apical surface and the basal surface. So this pathway in between the cells, this is known as the paracellular pathway, and only a very few selected material, just like a sodium ion, can move through this paracellular pathway, and it occludes everything else pretty much. Water cannot even move. It will be only moving through the aquaporin, through the cell, which is the transcellular pathway. So paracellular pathway is occluded by the tight junctions. This is number one. And the second function for this is that they actually compartmentalize the transporters. There are unique proteins which are found only in the apical surface, and there are some unique proteins that they are found only in the basolateral surface. For a very simple example, that if this is an epithelial cell, you find a sodium glucose co-transporter on the apical surface, which actually takes in sodium and the glucose into the cell. And then you are going to find on this side your GLU2 transporter, which actually moves the glucose from the cell into the bloodstream. And we just saw in a typical picture that in the basement membrane right here, you have the blood capillaries to which move these sugars after absorption. So composition of the plasma membrane is different. And you know that all the proteins actually move on the plasma membrane, but these protein or the transporters, due to the occluding junctions, cannot move from the apical surface to the basal surface or from the basolateral surface to the apical surface because they are being occluded. That's your occluding junction. So after that, we are looking at the adherence junction right here. So in this picture, if you see that these filaments are in red, you know that they are actins. So here you have one adherence junction and adherence junction. Now here, the word that is being used, that is essentially the general name. Remember that adherence junction, but they are on the lateral side of the cell. So they will be essentially the adherence junction, of course, due to the actin, but they are adhesion belts. They will make up the adhesion belts. And what is not shown in here, that we should also, we can just draw some structures right here, like an orange right here. We can just make some structures like orange right here. And then we can just use these red lines right here, and these will be the focal contacts. Now, the question is that you will ask that, remember that why the focal contacts has not been shown in this. Remember that the focal contacts will be present only in the cells which are motile, like the white blood cells. This is a cell which is sessile. It is fixed in one place. This is not going anywhere. And the focal contacts that's the reason that the focal contacts are not present in here. So this is just a hypothetical place that where the focal contacts will be. Here, the hemidesmosomes, which are the nuts and bolts, they actually keep these cells in this place. In this epithelial cells, there will be no focal contacts. They will be only in the cells which actually move over this matrix. And then what you have is your desmosomes, which are based with the blue colored intermediate filaments. On the lateral side, you have the desmosomes, which actually keep the two cells together. And in between the matrix, which is the basal lamina, and the basal surface of the cell, you have the hemidesmosomes. So these are all your anchoring junctions. Your two sets of here, it's usually one set, but you have the hemidesmosome, desmosomes, adherence junction. And if there is a cell which is motile, you will have a focal contacts instead of these adhesion belts. All right. So after that, what you have is also right here in between the cells, you are seeing that there are these gap junctions, which are made of these connection proteins. And we're going to talk about that. And that allows the movement of certain molecules of very small size. And those are essentially responsible for ionic communication, electrical communication in terms of ion movement. And we're going to see that. One of the important thing, that typical question that we ask, which of the following junctions are found on the lateral surface of an epithelial cells, or which of the following junctions are not found on the lateral surface of the cell? The same way we can ask this, like which of the thing are found basal layer of the cells and which are not found on the basal layer of the cells. This slides actually will summarize most of the detail of those in here. And here, what we are looking at is the same picture that maintain the polarity of the epithelial cells. Proteins from the apical surface, this green one cannot move in there. The typical answer, and say sometimes we ask this question like this way. Say, for an example, which of the following statements are true for the tight junctions? And say, for an example, one of them which are correct is that the tight junction seal the space between the cells to restrict the paracellular transport to only a few small molecules. So just remember that a few small molecules, but it restricts the movement of most through this paracellular pathway. So only a few selected molecules can be moved. And the tight junctions also fence off the plasma membrane domains to prevent the mixing of the apical and the basal surface protein. So it essentially fence off these green and the red proteins. So these are the two most important function with that. And say, for an example, on the other hand, if we just give you an option that which are the following are characteristic of the occluding or the tight junctions, and say that the tight junctions allow small molecules to pass from one cell to the other, or from the basal surface to the apical surface, that will be very wrong. So just make sure that you understand that differences in between that. And that will do it pretty much. And let's see what it is. So these are just some pictures of with some electron dense dyes that is added onto the apical surface, and then the pictures are being taken. So here you are seeing the junction, And these are individual, these finger-like microvilli from the cell. This is the typical surface of the cell. And you can see that electron-dense dye is on the apical surface. It cannot move through the paracellular pathway. So right here is your tight junctions. On the other hand, here, what we have seen that this is one cell on the left-hand side. This is the right-hand side. And right here is a tight junctions, and the dye was applied to the basal layer, and it cannot move past this tight junctions. So based on that, we have some question with that. So right here, you can see this lanthanum hydroxide is actually electron dense material, and this just shows you these experimentally, essentially. All right, so here we are looking at the occluding junction, tight junction exceptions. Allow epithelial cell sheets to serve as barrier to solute diffusion, and the paracellular pathway only allows only very few selected molecule or ions to pass, like sodium. So just know that what are the different pathways they are called. This is your paracellular pathway, which is occluded by the most molecule, and this is called the transcellular pathway. So most of the molecules, if they need to move in between the apical and the basal surface, they need to move either diffusing through the membrane, which are very few molecules like that is possible for them, and most of them are moved through some kind of carriers, which could be channels, pumps, symporters, or antiporters. And even for the water, you need if the water is moving from this area to this, you need to have a set of aquaporin from here, which aquaporin bring the water from apical surface into the cell, and then from an aquaporin that will move from the cell to the basolateral layer if the water is being absorbed by this layer. So these are some of the consideration that you can actually think of. All right, so we always revisit our occluding junctions, and we have always seen these as CFTR and the normal lungs. So right here, we are revisiting that. We talked about the tight junctions, and this sodium is the only exceptions that you can do. So essentially, if we just recapping the CFTR, what happens that right here is your CFTR through which the chloride is moved into the airway lumen. And as a negative charge is moving into the airway lumen, it actually pulls sodium to neutralize that charge. Sodium is in the blood side right here, much more. And this sodium is that exception that can move through the tight junctions. So now you have Na plus Cl minus in the airway lumen, which is sodium chloride that will draw water from the basal layer or the basolateral layer in here. And this is your transcellular pathway. See that sodium is going through the paracellular pathway, water is going through the transcellular pathway. It will cross through the aquaporin from here, from the capillaries of the basolateral interstitial fluid into the cell with one set of aquaporin, and another set of aquaporin will take it from the cell into the airway lumen, and they will dilute the mucus essentially, and that's normal lungs function. Whereas if you have impaired CFTR, you cannot move the chloride effectively into the airway lumen, and that will not pull the sodium, and you will not have a water drawn into that. So you will have a thick mucus, which can lead to infections and other. All right, so these are all very straightforward. And here we are looking at again, we talked about this. All right, so here also another picture, and this is the same picture. If you just think about that, this is the picture where you have the apical surface at the bottom. Now, this picture is now sideways. So if you just look at this picture now, the apical surface is on your right-hand side instead of the top. This is of your intestine. This is the hollow of the intestine. And here you have the glucose and the transporter. These are symporters, and they are taken up by the cell. Included, on the other hand, what you are seeing that here you have a GLUT2 transporter, which to the basolateral goes from the cell into the bloodstream. So, and here you have a sodium-potassium pump, but remember that although it has been shown that the sodium-potassium pump is on the other side, sodium-potassium pump actually do have an universal distribution. They are on all sides. It's not necessary. But here, the occluding function is very specifically maintained by this sodium glucose symporter, which is never found on this side. and the GLUT2 will be never found on the apical surface. These are essentially being occluded. All right, so this is very straightforward. We are going to ask some of these questions that what are the occluding individual proteins. So here you can see that there are some occludin and tricellulin. These are some of the proteins which does have membrane domains. Then there is a claudin 1, which is also those four transmembrane domains. Usually ask is that a tight junction is made up of which of the following proteins. So just know that it is made up of JAMs, junctional adhesion molecules, including and tricell, as well as the claudin. So these are the things, and here you can see the distribution of these proteins. So these 4 proteins, know them. Looking at the anchoring junctions, and here you have the actin as the cytoskeletal filaments, and we call this one as the adherence junction. And cell-to-cell adhesion belts, cell-to-cell focal contacts, and if there are intermediate filaments, no cell-to-cell desmosomes, cell-to-matrix hemidesmosomes. Very straightforward questions. What is what? You should not have any problem. So here we are looking at some of the pictures which are just a schematic of the same pictures that we have in here, just a little bit different way, and a couple of different. And that picture is essentially, if you just go back all the way up in here, and you will be able to correlate this anchoring junction summary with each of these things that what we are seeing here. So I will just go a little bit back and forth. So the first anchoring junctions, we are looking at that if they are made up of 2 different proteins. And right here, we are looking at the cytoskeletal filament. Right here is a cytoskeletal filament. And here you have an intracellular attachment protein. And here you have a transmembrane linker protein. So you have cell 1 on the left-hand side, cell 2 on the right-hand side. And the first thing that what you have is that you have a transmembrane linker protein. Then you have an intracellular attachment protein, then a cytoskeletal filament. Now let's go one at a time, and then I will show you that how you basically explain. So first, think about that the cytoskeletal, and we are looking only on the side, not on the basal layer. So first, we start with actin. So if this cytoskeletal filament is actin right here, then we are going to see that what are your intracellular attachment proteins right here, and what are your linker proteins. So let's go to that chart. So right here, we are looking at cell-to-cell anchoring junctions, and we are looking at these adhesion belts, which is in between the 2 cells. That is EK adherent. These are the green ones. These are your EK adherents right here. If this is the red ones are actin. And then if we just go back to the intracellular attachment protein, these are many, but these are the ones that are important, is catenins and the vinculins. Blue ones, which are the intracellular attachment protein, catenin and vinculins. And the extracellular ligand means attached, or the cell, the number 1 cell's EK adherent is attached to the second cell, neighboring cell's EK adherent. That is what is meant by the extracellular ligand. So cell 1's EK adherent is attached to the cell 2's EK adherent. Back and to the adherent of cell 1 is associated with EK adherent of the cell 2. Then you have the catenins and the vinculins, and these are actin. This is your number 1. Now, the same here is the association between the extracellular matrix as actin. So this is also still actin. And if they're actin, let's take a look at what we are looking for. So right here, cell matrix anchoring protein, and we call this now focal contacts because intracellular cytoskeletal attachment protein is actin. So it will have a transmembrane linker protein, but these will be integrin. So instead of EK adherent, this will be the integrin, that's green arrow-like structure. And then the proteins that they are essentially binding with are the ECM proteins or extracellular matrix protein, which is many different types. See, here it's E-cadherin and E-cadherin. They are the same. Here they are different, integrins and ECM proteins. And you have the intracellular attachment protein, which are catenin vinculin type, but they have different names. They are called talin and alpha actinin. So that's the difference. So let's go back to the picture right here. So right here, this is your integrin if this is actin, and this is, you will have alpha actinin right here. So these are your focal contacts. Now we are changing these to, instead of talking about the actins, now let's change this to intermediate filaments. If these are intermediate filaments, what are the different proteins? And we're going to see that. So if this is made up of intermediate filaments, we know that, and we will call this structure as a hemidesmosomes. When it was actin, this was adhesion belt, and these were focal contact. Now it will be desmosomes and the hemidesmosomes. And sometimes these intracellular attachment proteins are also known as plaques. So if you just look at the desmosomes, intracellular cytoskeletal protein is intermediate filament, and the transmembrane linker protein are the cadherins. They have some specific name based on the desmo. They are known as desmoglins or desmocollins. And the intracellular attachment protein are desmoplackins, placoglobin, and cadherins right here in the neighboring cell. So they are also the same, just like this. So these are your desmosomes. If they are on the between the cell and the matrix, hemidesmosomes, intermediate filament, here you have the integrins, same just like your actin integrins. And here you have the desmoplakin-like proteins, and the extracellular ligand are ECM proteins. So I will highly suggest that definitely keep a printed slide, this one handy, and if this will be available to you, and we just specifically ask where is what, and you should not have any trouble finding it out. Remember that it's a general structure. Depending upon what these red filaments are, whether they're actin or intermediate filaments, all these proteins will do that chart. All right. So after that, what we are looking at anchoring junctions, we have the cell-to-cell anchoring junctions. We are talking about a little bit, and right here you can see we talked about the tight junctions in between the cell. Then adherence junction, you have the desmosomes, and the gap junctions, you have all the pictures. That's what we've shown. All right. So after that, what we are looking at, this picture is still a little bit realistic in the sense that it does have this three-dimensional shape. These are three epithelial cells, and basal surface, lateral surface, these are the microvilli where the actin goes in there. And it just shows that why we call this as an adhesion belt. So you can see that if you just take a cross section of that, this is like a belt-like actin-like structure that goes around, and you have the associated protein in there. And what we talked about, that the transmembrane linker protein in between the cells in that space, these are your E-cadherins. E stands for the epithelial cadherins. And then you have the intracellular attachment proteins, catenin and the vinculins, which are associated. That's all you need. So the important thing is that in the adhesion belt, the actin-based motor protein, myosin 2, is also associated with that. And we'll see that it actually plays a tremendous role in the vertebrate development. So what you are seeing in here is a sheet of epithelial cells. And if we're just looking at a roundish embryo, we are looking at this cross section. So this is essentially the top. If we just look at the top of the embryo, this is the top, and we are looking at the cells which are right here, and we are looking at those cells right here. So so you have these cells, and you can see that these epithelial cells, they are all attached with this green line, and this green line is essentially the adhesion belt. So what happens is that when there is an appropriate signal or developmental signal is received, these motor proteins, which are the myosin 2s, they actually move and they tighten up these. So what you are seeing that in one area, these cells, the the adhesion belt actually become tightened up. And this is just, think about that. You have these loop purse or anything pouch with a thing, and then you just pull it and just it just come close together. So that's exactly what is happening. So if you just look at the cells in the middle, you can see that they are tapered towards the top. So the bottom is wider, whereas they're tapered towards the top, because right here, the adhesion belt is actually pulled together by the help of these myosin 2. Remember that that's the motor protein. And that right here, this is the region where you have the tightening happens, and the asymmetric shape of the cell basically invaginates this part. And eventually this whole thing will come off the ectoderm. And here you have a separate epithelial tube, which we called as a neural tube on the brain. Ask from there that formation of neural tube in embryos involve tightening of adhesion belts near the apical surface of the cells to taper them. That asymmetry causes the invagination of the epithelial sheet. Which of the following protein involved? And you know that it is the actin and the myosin 2, both are involved in there. So actin is for the adhesion belt and the myosin. So if the question is asked, which are the protein? Remember that along with the myosin 2, the adhesion belt is made up of actin. So both of them are involved in there. So just answer specifically that what is being asked. All right. So now we have cell to matrix anchoring junctions. And if these are the cells which are essentially motile, and these are mainly the white blood cells. So these are the focal adhesion. Intracellular attachment proteins are talin and alpha-actinin. And let's take a look at that, what happens. Right here, you can see that this is the direction of the movement. And we actually saw this somewhere else, and we are going to see this later as well. So these are all the focal adhesion or the focal contacts. And as the front part, which we call the lamellipodium, that is above the matrix, and it extends, and then there is a new adhesion. And at the back, there is an old adhesion that is broken. And we are going to see that a phosphorylation and dephosphorylation actually takes place in these places for the cell to move. So the functioning of each of these proteins, how they are regulated, we're going to see that how this actually happens. So we're going to see that broken and reformed during the cell movement. So know that how this essentially happens. So these are all very, very straightforward. So should not have any trouble with these. All right. So after that, we are looking at these intermediate filaments, which are desmosomes. And you can see in electron micrography, it looks like a cytoplasmic plaques. And here are proteins, which are desmoglein, desmocolin. These are cadherin-like, these plate-like structure, which are known as a plaque. That's why it's known as a placoglobin or desmoplakin. The plaque word is in both in here. And then you have the intermediate filaments, keratin in epithelial cell, desmin in heart muscle cell. Remember that why you have two different names in here. We already talked about that when we talked about the cytoskeletal filaments. For all the microfilaments, it is actin. And for microtubules, it is tubulin. You have alpha and the beta tubulins. That's made up of all the microtubules, and actin makes up all the microfilaments. Whereas for the intermediate filaments, depending upon what kind of cell it is, they are different proteins. So that is what is being shown in here. So these intermediate filaments are made up of keratin in epithelial cells. So this is one of the reasons that these are extremely strong. And that's one of the reasons that we have these layers of 30, 40, 50 layers of dead cells on the surface of our skin, 200, 300 layers on the palm or the sole of our foot. And they are all kept together. All these cells are dead, but they are all kept together due to these desmosomes and the hemidesmosomes. And they can be extremely strong, and the entire cell is filled up with the keratin, which is an abrasion-resistant protein. And remember that the hooves or the horses and the cows or the antlers of the deer and others, these are all made up of these dead epithelial cells, made up of keratin, and each of these cells are associated, like attached to each other by these desmosomes, the desmosomes in between the cells, not with the matrix. But this keeps the integrity of that dead cells, and the cells are filled with the keratin, can become very, very strong structures. All right, so these are very straightforward question. So sometimes we can ask, is the desmosomes are similar to the adhesion belts in some of the followings? And one of the similarities are that, remember that the transmembrane linker proteins are cadherins, and one of the answer choice could be that their transmembrane linker proteins are cadherin-like. So the transmembrane linker protein in the desmosomes are similar to the transmembrane linker proteins that what you have in your adhesion, and that will do it. So after that, this is just information that remember that what is pemphigus vulgaris, autoimmune disorder, autoantibodies against this desmoglin, which right here you can see that the desmoglin is one of the proteins which is in between the cell linker protein, cadherin-like protein. So the cells actually come off, blister, and only affect the skin desmosomes. So know that what the pemphigus vulgaris is. Now you have the hemidesmosomes, and here you have the integrins associated with the ECM. And we also ask hemidesmosomes are typical questions that are similar to focal adhesions in the following ways, that both use, say for an example, if we just give you the idea that both use integrin as the transmembrane linker protein, and both interact with extracellular matrix protein. These are similarities. On the other hand, if we just see that there's an answer choice that hemidesmosome and focal context both use intercellular cytoskeletal proteins, cytoskeletal attachment protein, intercellular attachment protein for the intermediate filaments, that will be different. So see that the intercellular attachment protein is desmoplakin-like proteins right here. And here in the focal context, the intercellular attachment protein is talin and actinin. So just make sure that you have this chart together, and this will actually help a lot. So here, what we are, I'm pointing out that what are the things similar. See, for desmosomes and hemidesmosomes, the intracellular attachment proteins are similar. Desmoplacoglobin, placoglobin. That's why they are colored the same. On the other hand, see that extracellular ligand between the adhesion belts and the desmosome, they are similar. That's why they are colored the same. And in between the focal context and hemidesmosomes, they are colored the same because the extracellular ligand is the same. Whereas you can see that transmembrane linker proteins between the focal contact hemidesmosome, they are both integrins. So based on this, we can have several questions. Just make sure you read the question correctly, and so that you don't miss that what is difficult. Compare similarity differences, compare adherence junction with desmosomes, focal contact with hemidesmosomes. This is basically very important for your exam. So that will do it. All right. So after that, we are moving into gap junctions. And here what we are looking at, that approximately molecules about 1,200 Daltons can actually cross, which is, remember that, which is just 1 kilodalton. And sometimes people miss that one. Instead of Dalton, if I just give this as 1 kilodalton, 100 kilodalton, 1,000 kilodalton, and 10,000 kilodalton, remember that 1 kilodalton is 1,000 Dalton. So that is important. KDA is 1,000 DA. So that is important. Just look at that. And inorganic ions, amino acid, nucleotides, vitamins can actually cross, whereas the protein, peptide, nucleic acid, polysaccharide cannot cross the gap junctions. One of the important things is that if you put a full fluorescent tracer molecule right here, so fluorescent tracer molecules are usually, say for an example, GFP. So if we just inject, say for an example, like a green fluorescence protein or red fluorescence protein, as soon as you see the word, you do not even have to know the molecular weight or anything like that. A typical question that we can ask is, say for example, we just injected green fluorescence protein right here, and after some times we just stimulated the cell. What will essentially happen? Suppose this cell was green. Remember that no matter what, that this will not be able to go through the gap junctions. Because, say for an example, GFP, green fluorescence protein, this is protein, and it will not pass through those. So any protein peptide. So if you just add those and the fluorescent tracer molecule is greater than 1000 Dalton, it will not pass. Most proteins are, so they will not be able to move through. So that's basically the typical question that you definitely remember. All right, so that will do it. And then what we are looking at is the structure. So what we're looking, you have cell 1 on the left-hand side, cell 2. If we actually look at this, the gap junction is made up of two halves. You have one half on this side and another half in this side. So this protein is known as the connexon. Right here is known as a connexon. And if we just take up the individual protein, one protein is made up of 1, 2, 3, 4 loops. So you have an N-terminal in the cytosolic side and a C-terminal also on the cytosolic side, and you have 4 transmembrane domains. So each of the connexon halves is made up of 6 of these connexins. So remember that connexin is the protein, and you have 1, 2, 3, 4, 5, 6. So this is this one is one connexin. So you have one connexin, another connexin, another connexin, another. So you have 6 connexin molecules that make half of this, which is the connexin. And in a total gap junction, you have one connexin, another connexin, 2 connexins. So you have altogether 6 connexin proteins on this side, 6 connexin proteins on this side, altogether 12 connexins. So just remember that. And another question that is asked in the, I think that it's one of the distractors. There is a protein known as inexin in your textbook, but I just wanted to just point it out. It's inexin. Instead of connexin, is inexin. And this inexin is insect protein. So it's an insect gap junction protein. But in all vertebrate, this is it's in the insect. So that's where the word comes from. This connexin, these are the proteins in all vertebrates. So that's basically but there is a word that is inexin. You may see that in one of the quizzes, vertebrates. Inexin in insects, but they also make the gap junctions where it was studied initially. Yep, all right. So now what you have is that how you regulate the closing and the opening of the gap junctions. In high calcium and low pH, the gap junctions closed. If you have a low calcium in the cell, that will open up the gap junction. And if you have a high pH in the cell, that will also open up the gap junction. Now, a typical question that sometimes we ask in association with that, suppose you have a cell right here, and then you have a cell right here, and you have a cell right here. And I have injected, say, for example, a green fluorescence protein into this cell. So this is all green right here. This is all green. I have injected green fluorescence protein. And then I saw that initially this cell was in a Petri plate, which does have a high calcium or low pH. Then what I do that after I injected the dye into the cell, I aspirate the media out. And in here, I just put a low calcium buffer with high pH. Either one of them will do. So either I say low calcium or high pH. So which of the following things are going to happen? So there will be a typical 2-3 pictures of the same. So this is a picture based. The dye will not move. I am trying to make you think that a low calcium will open up the gap junctions, which is correct. But remember that I injected a protein. No matter whether it is closed or open, it's not going anywhere. This picture, it will remain at this picture. So just make sure that we can just have this is a trick. So, and it will not pass through, whether closed or open, doesn't matter. So just remember that. So let's start with the part 2. All right. So now we are looking at the molecular mechanism of the adhesion. And first, we are looking at a cell-to-cell interaction and cell-to-cell to matrix. We're going to see that. And there are a couple of different types of cell-to-cell interaction. The first one is a homophilic binding. And we saw that just now in between the adhesion belts, that you have the cadherins in between your desmosomes, E-cadherin-like proteins as well. So that is your homophilic binding. We are going to see the heterophilic binding, where the two different types of cell, and the ones that what you also have is that binding to an extracellular linker molecule. And usually we look at these into the immune cells essentially, and we are not going to discuss that beyond the scope of that. And also see the interaction between the cell matrix, cell and the matrix, which is the ECM proteins. So let's take a look at that. What are the major families of cell adhesion molecules? The first one is your calcium, is your cadherins or E-cadherins based on epithelial cadherins. And you can see that each one of them remain as a dimer, interact with another set of dimers on the other side. And they have these calcium binding sites. So calcium is absolutely important with that. And then you have the Ig superfamily of CAMs, known as these sometimes NCAMs or neural cellular adhesion molecule. So neural CAM, NCAM, depending upon that, we're going to talk about that. And these are Ig or immunoglobulin superfamily. These are nothing to do with antibodies. But a typical structure is that there are some loops or the folds, which we see in, like, see that if you just think about the structure, then why we call this as Ig superfamily. See that if you look at the typical structure of antibodies, they are like this. And see that this fold right here is one of the arm like of these Ys right here. And this is essentially the typical structure of the heavy chain of the antibody. This is the typical structure of the light chain of the antibody. That is the reason that we call them as the Ig superfamily. It's a very large superfamily. There are 24 families of protein underneath that, hundreds of different proteins, and they have this typical structure, and you can see that they interact with that. Then we are going to see some heterophilic interaction, and we're going to see the integrins. Integrins is a heterodimer. So you have an alpha chain and a beta chain, and a combination of this alpha beta actually give them a large number of different types of integrins. So what type of alpha, what type of beta, that determines. And it also binds to a large number of ECM proteins. And here you are seeing one of them is a fibronectin, but there are many other, and these are all heterophilic interactions. Then we are looking at these selectins right here. Selectin is also bind to sugar glycoproteins. So this is another heterophilic interaction. And also, interestingly, that the integrins also can bind to the Ig superfamily. So you can see Ig superfamily can be involved in homophilic interaction. On the other hand, the Ig superfamily can interact with the integrin as well. So we are going to see each one of them. And what you are looking at, that see right here, there is a word called lectin domain right here. And the lectin is a sugar binding protein domain. So just remember that this is a whole sugar binding domain. And these sugars are usually most of the time mannose, but that is not needed, sugar binding domain. So this is what we called as a lectin domain. Another thing is that when we talk about this, we can use this word instead of sugar. We can use the words carbohydrate. Remember that this is also another way. We are looking at the cadherins first and how the different ones, and we find them in the adherens junction and the desmosomes. And the cadherins are E-cadherins. They are, the word is epithelial cadherin, pre-implantation embryos, non-neural epithelial tissue, and from this epithelia, we call this E-cadherin. So the P-cadherin, the name is from the placental cadherin. They are in the trophoblast, the embryonic layer, and this epidermis. You see the P-cadherin. N-cadherin is a neural cadherin, nervous centers, lens, cardiac, and skeletal muscle. We see that. And also remember that desmoglin and desmocolin, which are found in the desmosomes, they are also a type of cadherin, and these are all homophilic interaction, means both of them are the same type. So what is a typical structure? Now you can see that there is an extracellular domain, and you have the N-terminal outside, and then there are 3-4 calcium binding domains, and each domain has about 100 amino acid, and so each one of them. And then you have transmembrane domain, and then you have a cytosolic domain, and the cytosolic domain binds with the plaque proteins or the intracellular attachment proteins. These are catenins and the vinculin, which will bind to the actin. So if you just know this structure, that will be helpful. So this is a very simplified structure. There is a detailed structure from the textbook, and all the detailed name of each one of them is not necessary. If you just remember these, the ones, this will be more than enough. A typical question that what we ask, if we just compare this cadherin picture of this one with the integrin, see what happens that this has 2 chains. It's a dimer. This also has 2 chains. But as I said, that integrin diversity can be increased if you chain the alpha and a beta. So suppose here you have an alpha V and beta 3, or say if you have an alpha 1 and then you have a beta 1, this is one type of integrin. Then you can have an alpha 1 and a beta 2, it will be different. Then if you have an alpha 2 and a beta 1, that will be again a different one. So if you change the alpha and the beta chain, the integrin is going to get diverse. Based on that, and just jumping a lot of them, and then just showing you right here. See right here, alpha 1 beta 1, alpha 2 beta 1, alpha 3 beta 1, and this basically gives the integrins this diversity. On the other hand, what happens that for your cadherin, a homodimer, and you cannot change that. So if we just have a question is like, say, which of the following statements regarding cadherin is false? And if we just have option that cadherin diversity is expanded by the dimerization of a distinct chain type, that will be false. These are distinct chain type, dimerization of 2 distinct chain types. Here, dimerization of 2 distinct chain does not happen. Both of them are exactly the same. And they are homophilic attachment. Adherence depends upon presence of the calcium ions. These are correct, but they cannot be changed by the distinct chain types, unlike integrin. That's essentially your answers. This is a typical detail, and I just did not put a star in there because this is just a little bit more detail. And again, that detail is not necessary. If you just know the proteins right here, and which is included in the chart right here, that is good enough for your exam. So this is, but just know that there are many other proteins which can involve in this process as well. So this is a typical experiment. These are mouse L cells, and they do not express the cadherins. And you take these 3, so the experiment was done in the process that you take these 3 sets of L cells, and then you just transfect the vectors with the mRNA, which is a eMRNA, pMRNA, and the nMRNA. And they are just going into the cell, and they are going to produce the E cadherins, placental cadherins, and the neural cadherins. And then what you do then, so now you mix all of those, the E cadherin, P cadherin, and the N cadherins, and allow them to come together. And what you are going to see that as they are all homophilic interactions, what you are going to see that after sometimes by 8 hours, you are going to see cells where this is, although this is a black and white picture, but you are going to see that in between the cellular junction, only one type of fluorescence you are going to see. What do we mean by that? If you have the GFP with the E cadherin, you are going to see green in between them. If the P cadherin is in red, you are not going to see green and red, or green or yellow or red. So that gives you the idea that there is only homophilic interaction. What happens? So E and E interacts with together, the PP interact with together, and N interact with together. P and N will not interact. All of them are homophilic interaction. So a typical question that sometimes we can ask, we just take a part of an embryo, and then we are dissociated. We dissociate it, And then the cells initially sort by associating with other cells of the same type. And then we just give you a picture. You have all these different cells. So what do we mean by that? Let me just draw in a crude way right here. Break up these cells. You break them up, and then you allow them to associate. This is like a hypothetical picture. Remember that this is a totally hypothetical picture. So you have these blues, and then you have the reds. And then you just mix them up. They look like this. Like there, you break them up. This is temporary. This is temporary. And then finally, what you are going to see, you are again going to see that all these reds are in the middle right here. And it's reconstituted just like this initial picture. So all these blues are on the outside. And inside you have the reds. This is mixed up. And if we just give you that experiment visually and ask you that which essentially cell adhesion molecule is responsible for this, and your simple answer will be the cadherin. And that's essentially that's all. Very straightforward question. Just a visual representation. We can give you a visual representation, because remember that right here, if you see that pre-implantation embryos, trophoblast, placenta. So all of these association of these cells depend upon all the cadherins. So these are the reasons that just remember that whenever you see the embryo breaking up, coming together, it is all the function of these cadherins, and it's a homophilic interaction. Remember that. Same cells will come together at the same place. All right, so after that, we are looking at these Ig superfamily of the CAMs. Now, these are essentially the typical folds. They come in different types. So some could be soluble, and we're not going to talk about that. This is a GPI anchored protein right here. And then you have the transmembrane domain, where the cytosolic domain can be very long. And what we mean by these typical folds of the Ig folds, that in between these, this red, these are two S. These are two disulfide bonds. So in S and S. So these are two S's, and these are the disulfide bonds, which gives them this typical structure of these protein or the folds. And that is what it is. So independent homophilic interaction, remember that cadherin is calcium dependent, whereas Ig superfamily is calcium independent. Heterophilic interactions with integrin, and these are the neural CAMs in nerve cells and ICAM in endothelial cells. The I comes from the interstitial, that word, ICAM1, ICAM2. And we are going to talk about this ICAM1 and ICAM2 a lot, and in the extravasation later. And we'll see that this Ig superfamily, these proteins will interact with the integrins in that part. Then we're going into the heterophilic interactions. This is integrin, extracellular matrix, ACM interaction. So first, taking a look at the structure of the integrin, you have an alpha chain, and make sure that you understand this very careful. So the alpha chain is essentially made like a one protein, but a mature integrin is actually cleaved, and they are connected by a disulfide bond. And that's why this hinge is very flexible. Protein was essentially produced. If you have your N terminal on this side right here, then you have your C terminal on this. Now you have two pieces of protein. You have an N terminal in here and a C terminal right here. Then you have another N terminal right here. Just draw it in red. So you have an N terminal here, C terminal here, another N terminal here, and a C terminal here. So you have a protein 1 and a protein 2, but they are all together. They are joined with a disulfide bond. So what is the difference? In between the 1 and 2, instead of a peptide bond, remember that, which is a peptide bond, and remember that these peptide bonds are always a C to N, like a from your COH to the NH2, the CN bond or the peptide bond. Instead of a peptide bond, you have a disulfide bond. So that's why that they are not connected together as such. So what it gives, that this gives a little bit of flexibility of that, and this allows this area to work as a hinge, and this A sub alpha chain actually can bend considerably due to this difference, which wouldn't be possible if there is a peptide bond. So that's your alpha chain, and it does have several domains in its head, which either bind to calcium or magnesium ions for its function. It is essential that calcium, magnesium for its function that is required. Then you have the beta chain, that is a one straight one, but there is no gap in between N to C terminal end. And there is a middle neck region, which is rich in cysteine. And the beta chain bind to talin and alpha actin. This is in between lines. So just make sure that you underline that alpha chain do not bind to the intracellular protein. Only the beta chain binds to the talin and alpha actinin, and this is in between lines. It's very small and it's easy to miss. That's why I just made it bold. So just make sure that this is extra important. This is really in between lines. So definitely remember that. All right. So let's take a look at that, how the diversity of these varies. So you can see that it's a heterodimer alpha beta chain. And depending upon that, there are 1, 2, 3, 4 types. That's the number essentially. If it's a L, it's a large, M medium, sometimes is alpha 2B. There are many different types of names in there, and there are many cellular distribution, and you have all these different ligands. There are greater than 22. You do not have to memorize those. Just the one that we are going to talk about is this alpha L beta 2. This is found in a T cells or T lymphocytes, and we're going to see that they interact with ICAM1 and ICAM2, and we are going to see that this interaction in a second. And all leukocytes express with that alpha L chain and a beta 2 chain, which bind to ICAM and IQ. So just remember that this is not only the T lymphocyte, all leukocyte or white blood cells actually produce this. So that's why I just put this one: all leukocytes, all white blood cells. So just not T lymphocytes. All right. So on the other hand, we can just ask kind of like a philosophical question on that as well. That suppose a person who has a mutation where he cannot make this beta 2 chain right here, and what kind of disease or symptoms that person may suffer. So one of the important thing that we are going to see, that extravasation or essentially the white blood cells need to move from the blood capillaries into the tissue space for their functioning. And if you do not have this beta 2 chain functional on the leukocytes or the white blood cells essential for immune support, they are not going to move into the tissue space, and probably that person will be immunocompromised or suffer from some kind of immunodeficiency, genetic immunodeficiency. Any mutation of these alpha L or beta 2 chain on the leukocyte can lead to some kind of immunocompromisation or genetic immunodeficiency. All right. So after that, we are looking at that how the cells regulate the activity of the integrins. So here you can see that this is a regulation of matrix binding site. You have an extracellular matrix, and you have an inactive integrin, and you have an activating signal that the cells gets, okay, all right, we just need to essentially interact with this. And this activating signal binds to this particular intracellular signaling cascade, and this will trigger the integrin into a competent binding conformation. And now it is competent for binding. And if you just remember that at the very one of the slides, summary slides from your cellular signaling also actually had that many of these extracellular ligands, when they bind, they can activate the integrin is one of those functions. So this is an adhesion competent. Upon activation, it can bind. On the other hand, the regulation of cytoskeletal filament, that how they can be attached on the detached. So right here, you are seeing the extracellular matrix binding competent integrin, and now you have the actin cytoskeleton, and you have these attachment proteins right here. This is essentially a focal contact. Suppose it's a white blood cell. Now what happens if there is a phosphorylation of the integrin right here? Phosphorylation of the integrin happens. Immediately the cells will dissociate the attachment proteins from the actin, and it will dissociate from the actin cortex and disperse. So the serine residues on the beta chain of the integrins are phosphorylated. So where this is happening, just remember that this is an important. So this is this is a phosphorylation of integrin right here, phosphorylation. When they need to be activated, these phosphates needed to be removed. So they are needed to be desphosphorylated so that they can become binding competent. So from this slide, we will go back to this. So essentially what you are seeing that right here, you have a focal adhesion. This is one of the quiz question that I just put it in there, think about that right here, you have the focal adhesion, which is taken off from that. So this is a deadhesion. So what is happening in here? So here at the focal adhesion, the serine residues of beta chain are not phosphorylated. At the deadhesion, serine residues of beta chain integrin are phosphorylated. Signal transduction induce intercellular kinases to phosphorylate the integrins and deactivate them. All of the answer choice is correct. So just remember that the importance is that at the deadhesion focal point, when there is a deadhesion, you need to essentially phosphorylate that. Serine residues of the beta chain of the integrin are phosphorylated. That is the important part. And if you just look at that this is the focal adhesion. The serine residues on the beta chain integrins are not phosphorylated in any one of them. If they are phosphorylated, they will come off. Deadhesion, this is dephosphorylated, and the signal transduction induced intercellular kinases phosphorylate the integrin. So who will phosphorylate the integrin, which is not shown, that there are kinases, that is the extra part. Let's remember that. We learned that so I will just write it right here, of these phosphorylated protein are the kinase. Binding competent, this is also activation signal related, this is signal related, and this kinase, activated kinase, will phosphorylate the integrins, it will come off. And that being shown in the next slide. So just to remember that right here, that all of the answer choices are correct in this. So that is your correct answer. All right. So here, this is what is being shown, that the bending of this happens at the alpha chains with the disulfide bond, and you have this alpha and the beta domain. So just remember that this is your alpha chain and this is your beta chain. The beta chain also can bend to some extent because it does have this cysteine-rich region, and this is flexible, that can bend just like this disulfide bond. And what their structures are right here, the red one is an alpha, is the alpha chain, and it actually have a structure which is known as a beta propeller. And this is sometimes confusing. Just remember that the beta propeller has nothing to do with the beta chain. This is the beta chain, but the beta propeller is on the alpha chain, not on the beta chain. Why we call it a beta propeller? But you can see that you have these flat ribbon-like structures in blue. These are the flat beta sheets, and these flat beta sheets like structures right here, these blue beta sheet like structure, make up a flat round structure, and this is basically what we call as a beta propeller. And this is at the top of this the alpha chain, and you have a tapered domain on the top of the beta. So essentially what happens, that in an inactive integrin is in this bent format. It's in the bent format right here. So essentially, although this is schematic, just think about that. This integrin is in the bent format. When there's an activating signal comes in and the integrin is activated like this, it will actually stand up to interact with the corresponding the adhesion molecule, adhesion competent. So know this well. So model for integrin activation. So this is essentially the pathway that I was talking about. So here you can see that this is receptor tyrosine kinase. And we actually talked about this. This is the GRUB2, SOS, RAS, RAF, MEK MAP kinase, if you remember that, this is what we talked. But we do not need to remember all of these. But these receptor tyrosine kinase can also have various adapters and signaling kinases, which can activate integrins as well. And that can lead to cell proliferation, cytoskeletal organization, cell migration, gene transcription. All these things can cellular responses to adhesion receptor signaling. This can essentially happen. So that is serve as a signaling molecule by themselves. All right. So after that, we are looking at this last part is that selectins and the integrins. We're going to see this heterophilic interaction. And this is essentially the movement of the leukocytes. And we are going to spend the most of amount of time on this one. If you look at the blurb, it is a pretty detailed, and look at supplementary slides as well, because a lot of things happening in here. Know this well. So what we are looking at, this is the lining of the blood vessel. This is just only one side, and these are the flat cells are the lining of the blood vessels. Remember that it is, of course, completely schematic. If the blood vessels are like this, we are looking at just one of these, and we are looking from this side. So just one set of cells in there, just going back and forth. So that's what we are looking at. So what happens that in a normal condition, what you have that these are the blood capillaries, you have the vesicles where these P-selectins, where are the P-selectins? Remember that these are the lectin binding domain, which can bind to the sugars. They are already preformed through the protein synthesis pathway, and these are essentially the membrane bound protein. So you can see that this tiny tail is a membrane bound on the vesicle membrane, and these are synthesized through the secretory pathway. They are already produced and stored in the cell, but they are not on the surface of these epithelial cells. And the top portion is essentially the lumen of the blood vessels. So remember that. On the other hand, these cells have these other cell adhesion molecules, which are either ICAM1 right here, ICAM2. ICAM1 and 2 is present all the time, but P-selectins are not. And then we are looking at a leukocyte, and this could be many different types. This could be monocytes, this could be T lymphocytes, all these different types. What they do have on the surface, they have these alpha L beta 2 integrin. They are in bent conformation, which means that they are inactive. So they did not get any signal, inactive. And we're going to see that how the signal actually works. On the other hand, what you have right here on the surface, you have a selectin ligand, and these are specific carbohydrate. Selectin ligand. Where are the selectin? These are the selectins. And they are essentially for binding to these selectins. See that this is the carbohydrate, and see that right here, as I said, that instead of sugars, we can use this as carbohydrate. This is a glycoprotein. So essentially what you are looking at, this is the structure. This bottom part is the structure that you are seeing in the white blood cell leukocyte right here. This is what you are seeing in the leukocyte. So that is where people get confused. So in the next picture, this is what you have in the leukocyte. This is what you have in the leukocyte. I'm just writing leukocyte. This is what you have on the leukocytes. On the other hand, right here, this is what you have is on the epithelial cell. Is eventually, not yet, epithelial cells, and this is the epithelial cells lining the blood vessel. That is what you have in here. And let's take a look. And what you are going to see this, we talked about the ICAMs, which is the IG superfamily, and these are the ICAM 1 and 2. This is also the epithelial cells. So the pictures are a little bit confusing in the sense, here the bottom part is the white blood cells, the top part is the epithelial cell. Here the integrin part, this is the white blood cells or the leukocyte, and the bottom part is the epithelial cell. So that is what is being represented in the next picture. So now what is happening in here, that we got an inflammatory response. And essentially, there are several things that there are bradykinins and other molecules are involved. And there are many different other small chemical molecules that can be received on the cell surface receptors of these. Once they receive the signal, which is the inflammatory response from injured infected tissue cells, pathogens, what happens immediately, there is some signal transduction goes on into the cell that leads the p-selectins to be moved from the vessels on the surface. So these actually move all the way to the plasma membrane and they fuse with the plasma membrane. Now the p-selectins are on the surface of the plasma membrane along with the ICAM1 and the ICAM2, which were already there. And it does another thing. It actually expresses, which takes a little bit of time. This is a new protein, which was not there in here. So this is a new protein. This is a platelet activation factor. That protein is produced through the secretory pathway, and it is also moved into that. It takes a little bit of time, but it just moved into the surface. So two things happen due to the inflammatory response. P-selectin from the vessels moved to the membrane, platelet activation factor expressed and moved to the membrane. Now what happens that in a normal condition, there is no interaction with these white blood cells and these epithelial cells. So by the shearing force of the blood, they just move very, very fast. Now what happens, these p-selectins and these right here are the carbohydrates of the glycoproteins that are present on the white blood cells. They are sticky, but this is the importance. See, this is like a milder glue. They are not super tight. They are kind of sticky, tacky, but they are not super strong. This is what we mean by the low affinity. So what happens is the first adhesion interaction is a low affinity. Low affinity, just think about that. The low affinity means that this is a tacky, not a super strong glue. This is a milder glue. So the p-selectin and the ligand, which is the mucin-like, and causes the leukocyte to rolling as the low affinity interaction make and break. And another important thing is that the selectin ligand, that what you are seeing right here, this is mucin. So what is essentially mucin? Remember that this is your p-selectin. And as I said, that this is a glycoprotein. And this glycoprotein, the specific name that what you have for this one is mucin. So just make sure that nothing confuses you. That glycoprotein is your generic name, mucin with the sugar that is interacting, and this is mild glue. The shearing force of the blood, if it's just moving from this side, that essentially is pushing these white blood cells from left to right. So this will bind to another one and it will just roll and roll. So as it rolls, it slows down, but it is just not completely stopping it in one place. As it rolls, eventually, you can see that the leukocyte also do have a platelet activation factor receptor, or PAF receptor. This is already on the cell surface. But the platelet activation factor is only produced by the epithelial cells upon an inflammatory response. And as the leukocyte is rolling due to this mild first adhesion interaction, low affinity, they ultimately find this ligand and now they are engaged. So the PAF is bound to its PAF receptor. This actually triggers the signaling cascade, which is essentially a signaling cascade like this. And that signaling tradens the integrin. So the integrin was inactivated. Now see that PAF activates the integrin. They are now straight. So now what happens that this integrin and the ICAM1 and ICAM2, these interactions are the second adhesion interactions in high affinity. Integrin with ICAM1 or ICAM2 causes the leukocyte anchoring and pulling down due to the high affinity, followed by the extravasation. So if we just go to this picture back again, just remember that that this is high affinity, and we can just name this as a super glue instead of a mild glue. So this is much more stronger. That is what is happening. So at this time, when it interacts now with the integrin and the ICAM1 or ICAM2, it could be either the leukocyte is not going anywhere. It is going to sit on the epithelial cells, and the next portion, what is not being shown, that it actually flattens. So this is the most unique part. The entire actin remodeling happens into the leukocyte. This is not exactly like the pic. And the leukocyte flattens itself on the surface of the epithelial cells, left and right side, and finds the space in between the two epithelial cells. And the epithelial cells also undergoes a signal transduction, and it loosens up the actin, which is not being mentioned in here, but just the idea that in between the cells, remember that there are tight junctions. There are occluding tight junctions. Then you have the adhesion belts through which a small molecule cannot pass, how a cell can pass. So all those adhesions essentially break up. They needed to be completely loosened up, and that allows, without leaking any blood, this allows the leukocyte to squeeze through this into the subendothelial tissue, which is the tissue space. And this is essentially known as the extravasation. Leukocyte squeezes between the endothelial cell as it moves from the blood to the tissues. So that is the important. Remember that all these occluding and the anchoring junctions are broken up in specific places where the extravasation is taking place. And that essentially happens with all these signal transduction right here, cell migration and all those. These are cellular response for adhesion to receptor signaling, much complex receptor with this. And all these things happen due to that. And now it just goes in there. So just remember that integrins on leukocyte have one alpha chain and a beta chain, and each chain is a protein. That also remember that. And as I said, that if a person has a deficiency in alpha L chain production or beta 2 chain production, they will usually have less of these extravasation. So the leukocytes will not be able to interact with the ICAM, have extravasation, and essentially the person will suffer from immunocompromisation or genetic immunodeficiency as well. So that's pretty much the entire story. And that's pretty much about it. And let's see that we have one more slide. I guess I think that these are essentially all the summary of all of these, and that's it pretty much. So just shows all the different interactions, and now the interesting part is that this will tell you ultimately the whole thing, and we will talk about that much more in later. So in between the integrin and the extracellular matrix protein, this is the adhesion. Now, if you remember that we'll go in full circle with the first chapter, first few lines, that we remember that we use the EDTA to essentially chelate the calcium. And now you know that what is the reason that we just mopped up the calcium for taking the cells out, which are sticking on the surface of the Petri plates. Without the calcium, the integrin will not be functional. It will not be able to bind to the surface of those Petri plates, and they will come off. All these cellular, even the E-cadherins, these are also calcium binding. So when you take the calcium out, the CAMs or the cellular adhesion molecule will lose their functions, and that helps them to basically come loose. And then in the cancer biology, we'll see that that's essentially one of the reason that the cells actually can metastasize in other tissues. So that will do it.